Hello guys, welcome to IB360. Today's lecture will be on membrane transport. So previously we had talked about cell wall of eukaryotic cell and plasma membrane. Basically membrane transport is a part of plasma membrane, and membrane transport deals with passage of substances across biomembranes, mainly plasma membrane we will deal today. There are three classic methods by which membrane transport can occur: passive transport, active transport, and bulk transport. So, passive transport is the one which doesn't require external energy, where substances are flowing along their concentration gradient from their higher to lower concentration across our cell membrane. It can be broadly classified according to the substances which are following this particular transport into three categories. The first one is of neutral solutes and lipid soluble substances. So neutral solutes and fat soluble substances, they can move across plasma membrane through simple diffusion along the concentration gradient. That means from their side of higher concentration to the side of their lower concentration. And based on this particular feature, based on this free movement of lipid soluble substances across cell membrane, Overton in 1900 Overton in 1900, he proposed that cell membranes are made of lipids. So today the thing we know that cell membranes are a bilayer of phospholipids. We have studied the fluid mosaic model by Nicol and Sanger. But the first discovery about these lipids inside cell membrane were proposed by Overton in 1900, who gave this proposal mainly on the basis of this free movement of lipids across the bilayer actually. Then comes open channel transport. So open channels, they are not specific, they are always open, and these are mainly guided by the tunnel proteins. We have already talked about the tunnel proteins or transmembrane proteins, which are spanning throughout the bilayer. They are deeply embedded. So these proteins, they are always open to water mainly. So water channels or aquaporins are classic example of open channel transport, and the aquaporins, they allow water and water soluble gases like CO2 or O2 to move according to their concentration gradient. In fact, phenomena like filtration, ultrafiltration, they also follow similar mechanism. So what is the thing? The channels or the tunnel proteins, they're always open, and these are size specific but not solute specific or anything. In previous class I had mentioned that the cell membrane is semi permeable to water but selectively permeable to solute. So that we can understand now in intricate details why towards water it shows semi permeability. Okay, now comes ion channels. There are more than 100 ion channels that have been discovered, and ion channels are highly specific. These are highly specific, and there is a specific channel for each ion, specific towards each ion. And ion channels are also proteins, also which are also embedded in the membrane. Ions, they do not pass in dissolved state through ion channels, but instead only ions are moving through them. Because if they're in dissolved state, then they must be dissolved in some kind of solvent, which is usually water. But for water, we have already talked about aquaporins under open channel transport. which are different. Okay, so most ion channels are gated. Most ion channels are gated, and based on the stimulus required for opening the gates of ion channels, there are four types: voltage gated, mechanical gated, temperature gated, and ligand gated. But all this is again along concentration gradients, so obviously included under passive transport. Okay, so under passive transport, we have studied neutral solutes or lipid soluble substances which can get transported across the membrane through simple diffusion. They will follow simple diffusion. And from this, Overton in 1900 proposed that cell membranes must be made of lipids. That's why lipid soluble substances can so easily uh, move across the cell membrane through simple diffusion, because lipid soluble substances, they are also hydrophobic, and the inner environment of the cell membrane is also hydrophobic. Then comes open channel transport. They are always open, and classic examples are aquaporins, which facilitate or which allow movement of water and water soluble gases in the process across the cell membrane. Comes ion channels, which are highly specific. More than 100 ion channels have been already discovered, and they are specific towards each ion. And depending upon the stimuli which are required for opening gates, they can be voltage gated, they can be mechanical gated, they can be temperature gated, or ligand gated. Now comes active transport. Active transport is the movement of materials across the membrane where solute particles move against their chemical concentration or electrochemical gradient. So the movement is definitely against their concentration or electrical gradient. So there must be some kind of a gradient opposing to which the movement is taking place. It can be a chemical gradient, it can be an electrical gradient, and that is why energy is required for the process, because this is occurring against the usual flow, against the spontaneity, and this energy is obtained from ATP. Active transport occurs in in both ions as well as non-electrolytes. So ions and non-electrolytes, both of them will show active transport. Okay, non-electrolytes, like we can say salt, non-electrolytes with like glucose, then phenolphthalein in case of renal tubules, which undergo active transport. Now, the proteins which are involved in active transport, they are the carrier proteins. And why these are carrier proteins? Because they're showing saturation kinetics. What is saturation kinetics? Let us understand. So, saturation kinetics mainly is the rate of transport. That means the way the carrier proteins are transferring the solutes from ECF to ICF, they will increase with increasing solute concentration till a maximum is achieved. So it is almost like this. So even after increasing the solute concentration, the rate will be this, even after we increase. So this is solute concentration, and this is rate. So even so from up to this, you see up to this level, on increasing solute concentration, the rate is increasing. There is a constant increase in the rate in this region. But after a certain value, even though we are increasing the solute concentration, the rate is not increasing. It has achieved a maximum value, and this is saturation kinetics, which is displayed by the carrier proteins. So beyond this value, the rate of membrane transport does not increase, indicating that it takes place through the agency of these special organic molecules. And there is a special carrier molecule for each solute particle, ion or molecule. So they are also highly specific. And again, we already know that ATP is used in the process, and ATP gets dephosphorylated and turns into ADP, and this brings about certain conformational change. So as the solute particle, they are binding to the carrier protein, the carrier protein and also the ATP is binding. The carrier protein undergoes certain conformational changes, which ultimately results to some kind of movement, flip-flop movement, and that delivers the solute. It may be ion, it may be molecule, from ECF to ICF, and in the process ATP gets dephosphorylated. ATP gets converted to ADP, and this phosphate, which it is losing, inorganic phosphate, that is what is used to bring about the conformational change in the carrier protein. So both the solutes and the inorganic phosphate, which is coming from ATP, both are responsible for bringing about a certain conformational change in the carrier protein, which results in delivering the solute particles from ECF to ICF. This is the general mechanism of active transport, and obviously against concentration gradient that we have to understand, and saturation kinetics phenomena is also very important. Classic example is sodium potassium pump. Sodium potassium ATPase pump. It is an example of primary active transport. There is another thing called secondary active transport, but we are not delving into such details because they are not required for this level. Then comes bulk transport, the third part of our lecture today. So we have talked about passive transport, active transport, and now bulk transport. Now, for macromolecules which cannot undergo passive or active transport, that means which cannot cross the cell membrane through any kind of proteins, be it channels, transmembrane channels, or transmembrane carrier proteins, or even through simple diffusion, they are undergoing bulk transport, and it occurs by two methods: pinocytosis and phagocytosis. Pinocytosis and phagocytosis, they have similar mechanism, but a very important difference between the two is pinocytosis, in the nature of their solutes mainly. Pinocytosis mainly involves the uptake of fluid particles. So this process is often called cell drinking. It is the bulk transport of fluid matter and substances dissolved in it, like sugars, ions, amino acids. So they will transfer them across the cell membrane by forming minute detachable vesicles of 100 to 200 nanometer diameter. Cell drinking process. And another important term over here is detachable vesicles. Then comes phagocytosis. One more thing is solute intake may be selective or non-selective. So this may be selective or non-selective. Up till now, we have seen that in both for ion channels and carrier proteins in active transport, the proteins were highly selective, highly specific. For individual ions, there was an individual protein to transfer it across the membrane. But pinocytosis can be selective or non-selective. Depends on the solute, basically. And As soon as solute or ligand particles they form complexes with receptor sites, plasma membrane invaginates. This picture is of phagocytosis, but the mechanism is similar. So the plasma membrane will form an invagination like this, and the substance will be kind of swallowed in, and then the plasma membrane will be pinched off, something like this. So we can through our observation we can conclude one thing, that through both these processes, that is pinocytosis and phagocytosis, the circumference of the entire plasma membrane that is reducing, because a part of the plasma membrane is getting inside the cytoplasm by through formation of vesicles. Anyway, so as I was telling, the invagination depends and gets pinched off as a vesicle, and this vesicle would be called pinosome if it is pinocytosis, or phagosome if it is phagocytosis. So this pinosome will migrate towards the interior, and there it will liberate the material either in the cytoplasm, or it can be inside a vacuole. So for pinocytosis it will be pinosome, for phagocytosis phagosome. Phagocytosis is cell eating method, and it is the transfer of solid matter like food, foreign particles, pathogens, etc. across the membrane. Here also detachable vesicles would be formed. This is a diagram. First of all, you see binding and absorbing is done. WBCs, they undergo phagocytosis of foreign microbes. So first they will form this invagination of plasma membrane, then that will get pinched off, and this is the phagosome which is formed, phagosome. Now lysosome, how lysosome is involved in the process, you see. So as it is fusing with, this is a lysosome, as it is fusing with the lysosome, it is forming a digestive vacuole. So this is the digestive vacuole, which is also called phagolysosome, digestive vacuole. And then this will be digested, the microbe is digested, and process 4 marks the digestion thing, and then the microbial products, they are released outside. So this is how the phagocytosis is done. But if it is not a microbe, rather a solid particle, then the food will be digested, and the vacuole containing the indigestible substances like this, that will be called a residual vacuole. And the undigested parts are usually thrown out of the cell in the process of exocytosis called ephagy or cell vomiting. Ephagy or cell vomiting. So this is the term, ephagy. So this example is of phagocytosis by some WBCs as an important defense mechanism of the animal body. And every day almost 100 billion erythrocytes are destroyed, old erythrocytes, in the human body through this process, through phagocytosis in spleen and liver. So phagocytosis is cell eating. So these are enough informations about pinocytosis and phagocytosis. Hope you liked the video. Till then, stay tuned, subscribe our channel IB360, and stay safe. Thank you guys.